Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen, the participant of the 7th ICSAE, the International Seminar. My name is Tia Subositi, and in this forum, I would like to present our research entitled Genetic Characterization of ILER, Plectranthus Scutellariides based on RAPD molecular marker. ILER, Plectranthus scutellarioides, a herbaceous plant that has many variations in leaf character, growth habitus, and inflorescence characteristics. More than 500 cultivars of ILER were documented. Variation of leaves are consist of leaf shape, leaf color, margin shape, and foliar variegation. ILER is native to Africa and Southeast Asia and usually grown as an ornamental due to leaf variation. ILER is also used as a medicinal plant to treat newborns, dyspepsia, headache, asthma, and digestive problems. Based on clinical observation research on the efficacy and safety, ILER is one plant for hemorrhoids. Traditional herbal formula are known as jamu scientific to treat hemorrhoids. In view of its medicinal utilization for jamu scientific, and in order to ensure the quality of its supply, standardization process is usually needed. ILER standardization deals with its intraspecific variation, also adulteration or substitution among species in the genus. RAPD is one of molecular technique that has been successfully used to study genetic diversity and authentication in plant species. RAPD has several advantages due to not affected by age, physiological condition, and environmental factors. Also, it's low cost, rapid, easy, needs a small sample with no prior sequence information. The objective of this research was to determine of genetic characteristics among accessions of ILER Plectranthus scutellarioides based on RAPD molecular markers. A total of 15 ILER accessions and 3 accessions as an outgroup from genus Plectranthus, namely Plectranthus amboinicus, Plectranthus forsterii, Plectranthus forsterii variegated leaves were used as the samples. Accessions of ILER were collected from different locations, Java, Sumatra, and Papua, including ILER that cultivated in Tawangmangu. Those ILER accession has variation in leaf morphology, including leaf shape, leaf color, leaf margin, and flower color. Figure 1a is a ILER accession collected from Medan or Sumatra with the code MD1 to MD3, TW1 and TW2 from Tawangmangu. Figure 1b show ILER accessions from Tawangmangu with the code TW3 118H 11UHK and ILER collected from Malang, East Java with the code ML1 and ML2. Also, accessions GP collected from Jayapura. Figure 1c show ILER accessions from Tawangmangu, namely 118UHL, 118UHV, and 11UHUK. 3 species were used as an outgroup due to its morphological similarity with the code PA Plectranthus amboinicus, PFL Plectranthus forsterii variegata, PFP Plectranthus forsterii. Total genomic DNA was isolated from 100 mg fresh leaves using DNA kit isolation. The quality and quantity of extracted genomic DNA were determined using a spectrophotometry method and also in electrophoresis using 1% agarose gel. 6 RAPD primers were selected from 25 primers that produce clear and high polymorphism DNA fragments. Component of PCR reaction as follow, and PCR amplification were performed using thermal cycler with the following program. Amplified products were separated on 2% agarose gel. Only clear DNA fragments of PCR product were scored as presence or absence of each primer used and in all accessions. Similarity indexes of all accessions were calculated based on dissimilarity. The dendrogram was constructed using unweighted pair group method using arithmetic mean cluster analysis. This data analysis was performed by Anthesis version 2.0. Figure 2 shows total genomic DNA of 15 ILER accession and Plectranthus species. There was a good quality and quantity of genomic DNA in each accession. Out of 25 primers screened, 6 primers produce a clear and high polymorphism DNA fragments among accessions. A total of 52 amplified fragments were generated using 6 selected primers, of which 49 were polymorphic and 94.23% of polymorphism. Figure 3a shows RAPD profile of 15 ILER accession and Plectranthus species as a node group using RAPD primers. OPA1 generated 9 fragments and OPA18 generated 8 fragments. Figure 3b shows RAPD profile of 15 ILER accession and Plectranthus using RAPD primers OPB1 and OPD1. This primer generated 10 DNA fragments. Figure 3c shows RAPD profile of 15 ILER accession and Plectranthus using primers OPD2 that produce 9 fragments and OPH13 produce the lowest fragments, only 6 fragments with 100% of polymorphism. Table 1 shows primer OPB1 and OPC2 generated the highest number of amplified fragments. Meanwhile, primer OPA1, OPD2, and OPH13 show the highest polymorphism or 100% of polymorphism percentage. Dendrogram showed two major cluster groups that separated ILER accession and its outgroup with similarity index 40.45%. Index similarity among accession ranged from 82 to 97.87%, indicating low genetic diversity of Plectranthus scutellarioides. GP accession and 118H share the closest genetic relationship with 97.87% genetic similarity index. Cluster 1 not revealed for grouping 15 accessions based on origin of sample location. It was possible that the grouping based on morphological characters such as shape and color of leaves. Amplification using 6 RAPD primers also showed information on specific bands for authentication Plectranthus scutellarioides, Plectranthus amboinicus, and Plectranthus forsterii. Primer OPD2 only produced 2 specific fragments with a size 1050 base pairs and 1166 base pair. Meanwhile, the highest number, or 5 fragments, of specific fragments was amplified using the OPB1 primer, namely 5 band with a size 315 to 1159 base pairs. Those specific DNA fragments were generating in all accession of ILER and its species of 2 Plectranthus species. Screening and selection of specific DNA fragment based on reproducible in amplification of ILER accession and diode groups, so it's possible for ILER or Plectranthus scutellarioides authentication. There were 3 points of our research conclusion. 1. RAPD molecular markers were used for assessing genetic characterization of Plectranthus scutellarioides accessions. 2. 6 selected primer RIPD were revealed specific DNA fragment for authentication of Plectranthus scutellarioides among its potential adulterant, namely Plectranthus amboinicus and Plectranthus forsterii. 3. The genetic similarity index of Plectranthus scutellarioides ranged from 82.808 to 97.87%, indicating a low level of genetic diversity among accessions. This is the references that we use in our research. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.